Let's begin. So, you can always learn a lesson from studying biology and studying the microbial world. And I guess you can always learn, what would the word be? A new thesis or a lesson. There's always a life lesson in some random scientific study. So with this, I'm going to go over this particular paper, and then I'll just inject some of my thoughts. And this isn't a paper; this is a press release on phys.org. But regardless, I will have references to the citation in the description. So some bacteria sacrifice themselves to protect their brethren from antibiotics. So scientists at the University of Texas at Austin have discovered how some cells within a bacterial swarm will sacrifice themselves so that other cells in the swarm have a better chance of surviving onslaught by antibiotics. So the way that a lot of antibiotics work is that they will interact somehow with the bacteria, with one of the proteins or one of the biomolecules of the bacteria, and by interacting with that protein, it is able to mess up the metabolism of that particular bacterium. And so when these antibiotics are introduced and kill some of the bacteria will probably kill themselves a little bit more quickly than what you would normally expect, right? And this self-sacrifice is actually convincing some of the other bacteria nearby that they need to adapt their behavior for some sort of threat. So, and this is in a discovery important for the efforts to address antibiotic resistance. So this is a huge problem right now, especially in the context of, well, I would say in the context of STDs that are gaining more antibiotic resistance. It could make it more and more difficult to do treatment of certain types of STDs, but not just STDs. There's also issues with diseases that you can find in hospitals. So after someone undergoes surgery or undergoes just a routine procedure, if they end up catching an infection with antibiotic resistance, which is the result of being in such a sterilized environment, that I should just say that this is a field of study that's going to really need some revamping soon. So, as bacterial cells within a cluster or swarm die, they release chemical death cries, which scientists called necro signaling. So let's just do a quick search as, you know what, I don't think I see. Yeah, there's no Wikipedia article about it, so I won't go over it yet. So these signals act like a kind of early warning system, allowing the surviving bacterial cells to repair a type of resistance to antibiotics. The process of necrosignaling is outlined in a new paper out today in Nature Communications. So, bacterial swarms occur when certain types of bacteria, such as Escherichia coli or E. coli, band together by the billions and use their whip-like flagella to move as one over a solid surface. So all of these bacteria are moving somewhat or in together, I should say. So scientists have previously observed that these swarms are more resistant to antibiotics and knew that dead bacteria provide nutrients to surviving bacteria in the swarm. However, this is the first time an active signal has been uncovered. So Rasika Harshey, the Lorraine Morrow Kelly Professor of Microbiology, and her team had observed almost a decade ago that when bacterial swarms moved into an area that was treated with antibiotics, so remember, this is when you have this entire large cluster of bacteria all moving in synchronously, they move into an area with antibiotics present. And about 25% of the cells in the swarm will die with whatever concentration of antibiotic they used. And they wondered if this massive cell death was altruistic in actually helping the community as a whole survive. The results in the paper out today showed that this is in fact true. As they died, the cells released a protein that would bind to the surface of the surviving bacterial cells. This acted like a signal, letting survivors in the bacterial swarm know to start pumping the antibiotic out of the cells using specialized molecular machines called efflux pumps. So what are efflux pumps? So these are proteins that are on the cell membrane of the organisms, and what they do is, there isn't a good photo here, so just know that this would be an example of a type of protein pump or a type of pump, and this is going to use energy, use probably ATP or some form of energy currency within the cell in order to pump out antibiotics from within the cell to the outside of the cell. And but these take a lot of energy, right? So it's not good to have these running all the time, and probably they are inefficient, right? They probably also will pump out things that are actually important metabolites of the bacteria, and so you don't want these to be running on high gear all the time. However, when you are in the presence of an antibiotic, you as a bacteria will want to pump them out. And so you are going to invest the energy into pumping them out. And so when some of the when some of the bacteria die, these dead bacteria are releasing a signal that is warning all the surviving bacteria, hey, you need to start pumping this stuff out so you survive longer, right? And as a result of that, there will also be some extra nutrients available because you just lost a fourth of your population. So all the nutrients associated with those dead bacteria are going to be floating around in solution. So they probably have the resources to be able to do this pumping. So while this resistance is physiological, it buys the bacteria time to acquire mutations that would eventually lead to genetic resistance. So this is an extremely important thing to worry about. So, for instance, when you are told or when you are given a round of antibiotics, you need to finish your round of antibiotics, because if you don't, you are giving the bacteria more time to learn resistance to whatever antibiotic you were given. So you need to kill bacteria, I shouldn't say precisely, but with determination, and absolutely. You need to kill completely, cut off the bacteria strains that are killing you completely. Otherwise, you have just somewhat killed them, but then you have given them the incentive to learn antibiotic resistance without fully killing the entire colony. So this strategy that the bacteria are using when they are in these clusters, they are buying time for the surviving bacteria. So some of them will kill themselves off, which buys time for the surviving bacteria to eventually mutate and learn better strategies at surviving with the antibiotic around. So remember what I said, that using these efflux pumps or efflux pumps it requires a lot of energy to push them out push the metabolites or push the antibiotics out into the extracellular matrix, or the extracellular, just outside the cell, right? So that is inefficient. So if they can mutate a better way at surviving with antibiotics present, then that is going to take over and take effect. And then they won't have to be pumping out all these antibiotics, because they can live harmoniously with the antibiotics in the first place. So that is a problem. And if we can address this, we might be able to make it more difficult for the bacteria to mutate and to learn how to gain antibiotic resistance in the first place. So antibiotic-resistant bacteria represent a critical problem in medicine, because once a bacterium becomes resistant to several types of antibiotic medications, it becomes increasingly difficult to treat infections in humans and animals. So by understanding just one of the mechanisms by which these hard-to-kill swarms survive contact with antibiotics, scientists may be able to target that process with a therapeutic drug. So if we can find a drug when some of the cells decide to die and they release their signal out into the environment, if we can find a drug that will bind to that signal and prevent that signal from binding to the surfaces of the surviving bacteria, maybe we can stop this process from happening and end up killing more and more of the cells. I mean, that is a possibility. So Harshey and her team discovered through their research that within the bacterial swarm, a subpopulation of cells appear to be more susceptible to antibiotics, like a kind of bacterial cannon fodder. Or, I would say it's sort of like having canaries in the coal mine, right? So if you would always have this highly susceptible population, if this highly susceptible population dies immediately, that could be a warning sign that, hey, something is not right. You need to protect yourselves. So maybe these bacteria, they realize, oh, it's only they might say, oh yeah, the weak among us are dying, but so much of the weak among us are dying that maybe there is a problem in the environment, and therefore we need to build up our fortresses, if, for lack of a better word, or we need to be mindful of the environment that we are in because it's not conducive for our kind at this moment. So, by developing these different types of cells, the swarm has a better chance of survival than single bacterial cells. So, many clinically important bacteria go through phases of their infection cycle outside the host in harsh environments where swarming would increase their chances of acquiring antibiotic resistance through necro signaling, Harshey said. With better understanding of this process, we may be able to halt it. So what I want to also highlight is that we can learn from these bacterial colonies. So evolution is very, there's a lot of rhyming, right? There's a lot of rhyme and reason between different types of biological strategies that are employed from the microscopic level to the macroscopic level that we experience as humans. So, for instance, in the context of a pandemic, when we have a lot of what we would consider people with pre-existing conditions or people who are weaker in general, if a lot of them are dying, right, that means that there's something in the environment that is killing our kind. It is killing our species. And so it is very important as a entity or a unit of that species to look around you and say, there is a lot of us dying. I really need to care about what signals I am getting in my environment that is associated or correlated with all of this death. And even if the people that are dying are the weaker among us, or the old among us, or even the young among us. So, like, it doesn't necessarily have to be old people that are weak. Children are weak as well. So there are different types of diseases that go around. Regardless, all of this stuff that is happening around me, I need to take that in and then be put on high alert because the environment is now attacking me. And so I need to be on defense mode. This is the workings of a successful and healthy society. A healthy, altruistic society is going to see these signals and then say, hey, we need to go into a completely different mode or completely different thought process, or our mode should change so that we can survive whatever this new threat is. So that's one of the things that I can see here. One of the, I guess, symbolisms that you could, or connections, or homologies that you can make between our society and these bacterial colonies that are more successful at surviving than the individual unit. Now, something else that we really need to consider is that your own body probably does very similar things as well. As when you have some of your cells killing themselves, undergoing apoptosis or necrosis, they are going to release signals to the surrounding cells that help tell the surrounding cells what they need to do in order to survive, in order to repair the tissue, and to just be aware that there is a threat, there is something that killed me as a cell, and therefore all the surrounding cells need to get the signals to accurately alter their metabolism so that they can figure out what to do. So I think that this was extremely interesting to see a concrete example of how bacteria within the same species, even though the individual bacteria, we don't usually think of bacteria as multicellular because they're not. They are single cellular and they can survive by themselves. However, we really need to put more, we need to think more and meditate more on how even individual bacteria are able to function sort of multicellularly. They are able to emit and influence the environment and influence other bacteria around them. And so they are all sending signals to each other. Well, that's very similar to your own cells. Your own cells are just sending signals to each other. And so, I mean, what is the difference? Like, it's pretty much the same thing that is taking place. So with that, I'm at 15 minutes, so I think that's good enough for today. But sorry for the break, all 16 subscribers of you. But hopefully I will see you tomorrow.